In conclusion, we need to understand the impurity profiles in drug substances. And 2D NMR is a very powerful tool, which is also labor and time efficient, which allows us to do the structural elucidation. The workflow of the structural elucidation is quite simple and was demonstrated here. In most cases, the impurity structure can be completely resolved by using data from HSQC, quasi HSMBC. In some cases, you need to go to the higher level two-dimensional NMR, but it's quite rare in our practice. And again, I would stress that our knowledge of the chemistry, our logic is very important here, and also some additional very inexpensive analytical information coming from mass spectra and FTIR. NMR prediction also is very important because it allows us to select the right structure from the number of several of them. At the end, I would like to thank Dr. Louis Glunz, our founder, who is inspiration in this company for all of us, and Louis Glunz for his attention to the structural elucidation and to the pharma services, and in general, the company ownership for taking care of the business. Dr. Oscar Rodriguez, who actually isolated impurities A and B and provided them for structural elucidation. Our project managers, Sanjay Tyre and Stephanie Wu, who took care of the projects and got permission from the customers to disclose those structures. David Tsao, who is our NMR chemist and who is taking good care of the NMR instrument. And also Andy Miles, who is representative for pharma services from the business side. And you can contact him if you would like to get those services for your company. And if you have any questions regarding these presentations, you can also contact me. And this is the internet address. Thank you so much for your attention, and I would like to answer your questions if you have any. Yes.
So you ran all those spectra here?
Yes, those spectra which were presented were run on our 400 NMR system. If you need to run a higher level NMR, let's say inadequate, it would take too much time probably. It's more economically feasible to outsource inadequate. But those three types of NMR, you can run in the matter of 4 hours on our NMR system. Any other questions?
So how much sample you typically use?
In order to save time, it's good to use like 100 milligram samples. So for this particular case studies, it was used around 90 to 100 milligram. Because typically chemists have it. But I would say anything from 10 to 30 milligram would be sufficient. But it would take much more time.
How pure does the impurity have to be in order to get a good spectrum? Typically, 90%, 95%?
There are two approaches. Either you can purify the impurity to the level which is 80 to 95%, or you can have 2-3 samples which represent less purified material where the other part is variable. Let's say you have a sample which has 50% of your desired product, maybe 60 and 70, and it would be sufficient, because then you can see dynamics and you can identify the signals which belong to your compound and discard the signals from other impurities. So in the best case scenario, you purify it to above 80%. If you're unable to do it, you can collect fractions which contain variable amounts, but you have to see dynamics to elucidate the structures from your compound.
Did you do also the elemental analysis?
It's not necessary in most cases. Whatever you saw here was just done by using mass spectral data. And in these cases, because the molecules were quite simple, we didn't even use high-resolution mass spectra. But if you use high-resolution mass spectra, it would actually replace your elemental analysis because you will have a good clue on your molecular composition. Yes.
What other pieces of information you need from the customer to help you to count the structure? I mean, they submit you sample, what else you do?
Usually we would like to find out the chemistry where this compound was produced. Maybe not the all details, but the major details, where it was started, at what step it formed, The chemistry helps to make it more economically feasible. If you don't have this information, you will need to spend more time, and obviously the structural elucidation will be more expensive. Our customers typically provided this information, and we're able to do this work very economically and efficiently.
One question I had about the exchangeable protons, was that the information that you got from like IR? Like what kind of exchangeable protons you have? Was that from something like IR spectrum?
In the case where we were not able to find out exchangeable protons by NMR, we had to think about the molecular structure and obviously look at the FTR, because FTR showed that those heteroatoms, the oxygen and nitrogen, they should have hydrogen there.
Based upon the speculation from the starting material.
All right. And obviously NMR showed that somehow we have a couple of protons missing, so where they should be somewhere, because mass spectra shows they're here.
It wasn't like out of the blue, so based upon the predictions from the desired compound.
All right. But again, I would mention that FTRI is a very inexpensive method of analysis, and it's always good to have it alongside.
Igor, the NMR predictor that you used is very accurate. Most of them are just an estimate.
Because this estimate based on the empirical data from the thousands and tens of thousands of NMR spectra, it's quite accurate, even for the complicated structure. Again, if you saw, sometimes only a couple of signals are important. If you analyze lots of signals from the molecule, you'll see many of them are almost identical for the alternative structures, I mean. But you focus on those which are different. And this structure prediction typically gives you enough information. Any other questions?
Yeah, I just have one last question. So the table you showed us at the end, where you had all those linkages and everything, how long did it take you to arrive to that table from the starting point?
Well, you have to analyze your spectral data. Obviously, as I mentioned, to collect the spectral data would take probably half a day, NMR data, both one-dimensional and two-dimensional. And then it depends how complicated your structure. Now, this workflow, because you're doing everything stepwise, allow you to save time, because you're filling empty spaces in your table, and it may take a couple of days at the end still, because you have to, sometimes there's too many correlations from HMBC, and you have to think which should be discarded. Very often HMBC can be also fine-tuned to show correlations with 4 or 5 bonds even. And these are extremely important, and we have one case where we were able to elucidate the structure of one isomer versus another isomer using 5-bond correlation on HMBC spectrum, which is not typical, but it was very helpful in this case. Okay, if it's no other questions, I would like to thank you for your attention.